So good morning. I am Dr. A. Ranganath Reddy, Associate Professor, Department of Biotechnology, Vignan's Foundation for Science, Technology and Research. So today we'll be discussing about the topic is molecular modeling. So today we'll be discussing about this topic, molecular modeling. This subject in which the topic of our interest is Biosuite. So let us define this molecular model. So molecular modeling is also called as comparative modeling or homology modeling. Comparative modeling or homology modeling. So simple concept where we try to build a structure. We try to build a structure for a protein. So in this molecular modeling, it is also called as comparative modeling or homology modeling. We try to build a structure for a protein whose sequence is known and structure is unknown. So how we are going to build a structure for this protein whose sequence is known but structure is unknown. So here we are trying to compare the protein whose sequence is known with the proteins from the so-called database, that is SwissProt or UniProt KB. So basically this SwissProt and UniProt KB, they are protein sequence data. So here we have a protein whose sequence is known but structure is unknown. Here we are trying to build a structure for the protein. So in this process, we are trying to compare the protein whose sequence is known with the protein sequence data. So in this process, we are trying to find a match for our sequence protein from the database whose sequence and structure are already known. So here we are trying to build a structure for a protein of our interest whose sequence is known but structure is unknown by comparing it with a protein whose sequence and structure are already known from the database. So coming to this protein sequence, there are databases like SwissProt and then UniProt KB. And coming to the protein structure database, there is only one database, that is PDB. It stands for Protein Data Bank or Protein Database. So coming to the objective of this particular course, that is molecular modeling. So here the objective of this course is mainly to train students in utilizing basic molecular modeling techniques and to explore biological phenomena at the molecular level. So here the objective of this course is mainly to train the students in the field of molecular modeling. So as defined earlier, so molecular modeling is nothing but a technique where we are going to build a structure for a protein whose sequence is known but structure is unknown. How we are going to build the structure for that protein. So here the students will be trained on molecular modeling techniques and they are going to explore biological phenomena at the molecular level. Here the students also will be trained how to retrieve data from the database. Here the students are trained to retrieve data from the database. Once data is retrieved from the database, they are also trained on the molecular modeling tools. So once after retrieving the data from the database, they are going to apply the data to the modeling tools or modeling software. For example, Swiss Modeler and then EasyPrep 3D and then iTag. So these are the molecular modeling tools. Right. Coming to the outcome of this particular course, that is molecular modeling. Here, the student will be able to understand the principles and practice molecular modeling and drug discovery. So the outcome of this particular course, that is molecular modeling, is here the student will be able to understand the principle behind this molecular modeling. So what is the principle behind this molecular modeling? So he is well trained on this so-called molecular modeling technique, and he will also be trained how to retrieve the data from the database, and later he will also be trained how to apply the data which is retrieved from the database in this so-called molecular modeling software. And later, after this molecular modeling, the student next will proceed further to the next step, that is what we call as drug discovery. So once molecular modeling is done, the next step is nothing but drug discovery. So here the student not only understand the principles of this molecular modeling, but he will be made to practice this molecular modeling software. And once model is built, then the student is further taught about this drug discovery. So coming to the contents of this particular topic, here today we will be going about this Biosuite, and then what are the modules in the Biosuite, and finally we are going to conclude this in the form. So the first point here is we'll know about what is Biosuite, and next is what are all the modules in the Biosuite, and finally we are going to conclude this to summary. So first, what is a Biosuite? So what is the Biosuite? So Biosuite is nothing but a comprehensive bioinformatics software package. So Biosuite is nothing but comprehensive it is a bioinformatics software package. So Biosuite is a bioinformatics software package. It is a comprehensive software package which was developed by linking the industry and academia. Right. So Biosuite is a comprehensive bioinformatics software package which consists of many tools, and it is developed by linking the industry and then academia. It is developed by linking the industry and then the academia. So it is a joint collaboration between the industry, that is TCS, and then the academia people, and that led to the designing of comprehensive bioinformatics software package, and it is named as Biosuite. And the last decade has witnessed an exponential growth of information in the field of biological macromolecules. So here, what are the biological macromolecules? So basically, they are proteins, they may be nucleic acids, either they may be DNA or RNA, and then carbohydrates, right, and their interactions with other molecules. So here, let me define what is bioinformatics, and then why bioinformatics came into existence, right. So bioinformatics is defined as the application of IT, that is information technology, towards biology for the creation of biological database and development of computational tools. So bioinformatics will be defined as the application of information technology towards biology, which lead to the creation of biological database and development of computational tools. So the term bioinformatics was coined by Professor Pauline Fogweg, and bioinformatics came into limelight after the successful completion of Human Genome Project. So after the successful completion of Human Genome Project, the term bioinformatics came into limelight. And Human Genome Project was started in the year 1990 and it was completed in the year 2003. So it took almost 14 long years for successful completion of human genome sequence. So what is genome? So genome is defined as the totality of all the genes of a particular organism. So humans are roughly around 25,000 to 40,000 genes. So if you try to sequence the genes of a human, that like around 25,000 to 40,000 genes, that will give you the genome sequencing. So that is nothing but the genome sequence. So coming to this biological macromolecules, which mainly includes proteins, which is a polymer of amino acid, and then the nucleic acids. Basically, they are the genetic materials. The nucleic acids, again, there are two types. The first is DNA, which stands for deoxyribonucleic acid, and the second one is RNA, which stands for ribonucleic acid and carbohydrates. So these are all the examples of biological macromolecules. So why this biological database came into existence? So coming to this database, that is NCBI, which was started by Claude Pepper, the then senator, and this so-called Claude Pepper, he was very instrumental in designing this NCBI database, and which is maintained by NLM and then NIH, where NLM stands for National Library of Medicine and NIH stands for National Institute of Health. So what is the reason? What made Claude Pepper to design this NCBI database? As many people are working day in and day out in the labs, they end up with data at the end of every day. And if they start documenting all the data in books, then we need multi-story building to save the data. But instead, why can't we have a database where we can just store the data? So after creating a database is not only for storage of data, but once if we are storing the data in the database, so we can retrieve the data as and when it is required. So it is very easy to retrieve the data from the database, but whereas it is very tough to retrieve the data which is maintained in the records. So as the data is becoming robust, exponential growth is nothing but robustness. So as the data is becoming robust, right, and it is very difficult to store the data since the data is becoming robust. So they have designed this database, that is biological database. So biological database are meant for not only storage of data, but they are also meant for retrieving the data as and when it is required, right? And coming to this, computational analysis and predictions based on such information are increasingly becoming an essential and integral part of modern biology, right? So once we have the data with us, we are trying to save the data in the form of a database, and we are also developing computational tools, right? So with the data, whatever we have stored in the database, we can easily retrieve the data as and when it is required. And at the same time, we do have many computational tools. So after retrieving the data from the database, we can apply the data to the computational tools, which gives us many results. So Biosuite is nothing but a comprehensive bioinformatics software package which was developed, right, between the industry and academia. Here the industry and academia came together, right, as a joint venture. They have designed this Biosuite, and Biosuite is nothing but a comprehensive bioinformatics software package. Right. So in view of this exponential growth of the biological data, exponential growth which can be termed as robustness. As the data is becoming robust, or exponential growth of the biological data. In view of this, the CSIR, which stands for Council of Scientific and Industrial Research, India, it undertook an initiative to promote industry-academia collaboration. So who is instrumental in establishing this industry-academia collaboration? It is nothing but CSIR, Council of Scientific and Industrial Research, India. It was very instrumental, and it took an initiative to promote a unique industry-academia collaboration. Here you can see the collaboration between the industry and then the academia. Academia here includes universities, and they together they have developed a software package, right, under its New Millennium Initiative for Technology Leadership in India. The program, so they have initiated this Biosuite under the program which was named as New Millennium Initiative for Technology Leadership in India. So this is right. So CSIR and industry and academia, they came together and they formed as a collaboration, and they have developed this comprehensive bioinformatics software package which was named as Biosuite, right, under its New Millennium Initiative for Technology Leadership in India program. So under this program, they have designed this comprehensive bioinformatics software package which is called as Biosuite. So here the Biosuite, it integrates the functions of macromolecular sequence and structural analysis, cheminformatics, and algorithms for aiding drug discovery. Here, the data revolves around the biological macromolecules. So, as discussed in the previous slide, here the data revolves around the biological macromolecules. And here I would like to mention there are two types of sequences. The first one is nucleic acid sequence. The second one is amino acid sequence. So there are two types of sequences. First one is nucleic acid sequence, and the second one is amino acid sequence. Coming to this nucleic acid sequence, we can see the repetition of 4 nitrogen bases, that is adenine, guanine, cytosine, and then thymine. If it is a DNA, and adenine, guanine, cytosine, and then thymine is just replaced by URM. It is a RNA. And coming to the second type of sequence, that is amino acids, that is nothing but a protein. Protein is a polymer of amino acids, and we know there are 20 amino acids, and each amino acid is denoted by a single letter code. For example, P is the alphabet which codes for the amino acid glycine. Right. So Biosuite, which is a comprehensive bioinformatics software package, it integrates the functions of macromolecular sequence and structural analysis. So cheminformatics is nothing but a branch of science which deals with pharmacogenomics, and algorithms for aiding in drug discovery. So as mentioned earlier, after successful completion of this molecular modeling, next step is nothing but drug discovery. Right. So here this so-called Biosuite, it has 4 major modules. The first one is genome and proteome sequence analysis. The second module is 3D modeling and structural analysis. And the third module is molecular dynamic simulation. And fourth module is drug design. So here the Biosuite, it is mainly organized into 4 modules. So what are those 4 modules? The first one is genome and proteome sequence analysis, and the second module is 3D modeling and structural analysis, and the third module is molecular dynamics simulations, and fourth module, that is drug design. So this Biosuite, it is organized into 4 major modules. So the 4 modules are, the first one is genome and proteome sequence analysis. So here what is genome? So genome is defined as the totality of all the genes of an organism. So genome is defined as the totality of all the genes of an organism. Let us take for example human being, and the scientific name is Homo sapien. So human beings have roughly around 25,000 to 40,000 genes. So if you go for sequencing 25 to 40,000 genes of a human, then it constitutes the genome. And similarly, the next is nothing but proteome. So proteome is defined as the total protein content. So proteome is defined as total protein content of an organism. And second one is 3D modeling and structural analysis. That is the second module. And third module is regarding the dynamics and simulation part. And the fourth module, that is the last module, that is nothing but the drug design. So these are the 4 modules in this Biosuite. So here coming to this Biosuite, which is a software package developed by the collaboration between the industry and academia. This package, here the package refers to the Biosuite. Biosuite, it will be valuable for high quality academic research, industrial research and development, and for teaching purpose both locally within the country as well as in the international arena. As I mentioned earlier, Biosuite, it is mainly designed by the so-called TCS and then CSIR, which took an initiative to maintain a platform, to bring a platform between the industry and academia, and it resulted in the development of this Biosuite software package. And this package, it is of high importance in the field of academics, as academia is involved, and in the field of research, and in the field of industry, and it can also be used for teaching purpose, especially in the academia like engineering colleges, universities, both within the country as well as outside the country, that is in the international arena. So here the objective of this project, here the project refers to Biosuite. So here the objective of this project have been to develop indigenously a set of software tools, which would assist the academic research, R&D, research and development, and also application in industry in the rapidly emerging field of bioinformatics and drug design. So this Biosuite, which is a software package which consists of many tools which can be used by researchers and then academia as well as R&D, especially in the industry, and it is rapidly emerging field of bioinformatics and drug design. So as defined earlier, bioinformatics is an interdisciplinary field where you can see the application of information technology towards biological sciences, application of IT towards biological sciences, which lead to the creation of biological database and development of computational tools. So here the need for such a software suite, that is nothing but Biosuite, is exemplified by two main factors. So what are the two main factors which are very critical in the development of this so-called Biosuite, which is a package of software tools? So the first reason is increase in bioinformatics activities at all levels, which includes education sector, research, industry, and rapid growth of primary data and methods in computational biology. And the second thing is limitation of existing suites. So what are the two reasons behind the development of this Biosuite? The first reason is increase in the bioinformatic activities at all levels. So as mentioned earlier again, so after the successful completion of Human Genome Project, which was started in 89-90, and it was completed in 2003. Once this Human Genome Project was success, right, there bioinformatics gained a lot of importance. As a result, we are able to see the courses like BTech Bioinformatics, and then MTech Bioinformatics, and then PhD in Bioinformatics, and then PDF in the field of Bioinformatics. So, because of the successful completion of Human Genome Project, the bioinformatics came into limelight. As a result, we are able to see these courses like BTech Bioinformatics, MTech Bioinformatics, MSc Bioinformatics, PhD Bioinformatics, postgraduate PDF in Bioinformatics, and then we can also see various PG diploma in the field of Bioinformatics. So all this is because of the Human Genome Project. So because of success of Human Genome Project, bioinformatics came into limelight, which led to various courses which are being run by various universities. And at the same time, we can also see what is the importance of this bioinformatics, especially in the field of vaccine design. So the first and foremost step is nothing but sequencing the genome of a bacteria or a bacterial genome or viral genome. So the first step in this vaccine design or drug discovery is cracking the genome of the so-called bacteria or virus which is responsible for the infection, right? And at the same time, because of this robustness of the data, it led to the creation of biological databases. Even though biological databases were created before the starting of Human Genome Project, but the databases came into limelight only after the successful completion of Human Genome Project. So as of now, we have various types of databases in the field of bioinformatics which includes primary database, secondary database, and then structural database, biochemical database, and then literature database, and so on. So primary database mainly includes NCBI, DDBJ, and then EMBL, and secondary database mainly includes SwissProt and then PAR, and then structural database which mainly includes PDB. Coming to this biochemical database which mainly includes KEGG and Brenda, and so on. As of now, we are able to see many databases, and all these databases, they are in the public domain. So since they are in the public domain, we can just retrieve the data from the databases. And the second thing is the limitation of existing suits. So as of now, even before this Biosuite development, there were suits, but there was some limitation. So Biosuite was mainly designed to overcome the limitations of the existing suits. And here we can clearly see this Biosuite. So it consists of 4 important modules. The first one is sequence and genome analysis, that is the first module, and second module is the structural analysis, and third module is the simulation, and fourth module is the drug discovery. So if you club all these 4 modules, the first module which deals with sequence and genome analysis, and the second module which deals with structural analysis, and the third module which deals with the simulations, and fourth module which deals with the drug design. So these 4 modules together, they constitute the Biosuite. So coming to this first module, so first module is with respect to the sequence and then genome analysis. So coming to the sequence, we have basically 2 types of sequences. The first one is nucleic acid sequence, and the second one is the amino acid sequence. So basing on the types of sequences, so we do have databases, especially for nucleic acid sequence, we have NCBI, which stands for National Center for Biotechnology Information, and then DDBJ, which stands for DNA Data Bank of Japan, and third one is EMBL, which stands for European Molecular Biology Laboratory. So these 3, they come under the category of primary database. And coming to the second sequence, that is amino acid sequence, that which is nothing but protein. So we do have databases like SwissProt, and then UniProt KB, and then PIR, and then translated EMBL. So all these, they come under the category that is secondary database. So we have basically 2 types of sequences. First one is nucleic acid sequence, which can be retrieved from the database like NCBI, DDBJ, and EMBL. And coming to the second set of sequence, that is amino acid sequence, it has databases like SwissProt, UniProt KB, PAR, and then translated EMBL. So coming to this genome analysis. So genome is defined as the totality of all the genes of an organism. And if you would like to go for sequence, if you would like to go for analysis of the genome, it is nothing but Human Genome Project, right? So Human Genome Project, it mainly deals with the genome sequencing, that is genome analysis, that is genes, right? Similarly, so after the successful completion of Human Genome Project, so they have gone for sequencing the genome of many bacteria, many virus, many plants, and many other organisms. And coming to the second module, which is nothing but the structural analysis of the biological macromolecules. So we have a database that is PDB, which stands for Protein Data Bank or Protein Database, which is the structural database where we can find the structures of biological macromolecule, that is proteins. And we do have tools here for visualizing the 3D structures. So we do have tools in the field of bioinformatics which are mainly used for visualizing the 3-dimensional structure. For example, RASMOL, and then KHIME, CN3D, JMOL, MOL, and so on, right? And next thing is the simulations. So here we have tools for carrying out the simulations where we have NMD, VMD, and other tools, and we have ITASR, which is used for homology modeling, and later you can go for simulation studies. And here we do focus on energy minimization, where we have energy minimization tools like GROMACS, and then amber, and then chalk, right? And the last component is the drug design. Drug design, where we can perform this drug design by using Autodoc, Veena, and then gold, right? So these are few tools which are used for performing these docking studies. So docking is simply defined as the interaction between the receptor protein and then the ligand molecule, right? Docking is simply defined as the interaction between the receptor protein and then the ligand molecule. So and these ligand, it will be later converted as the drug, right? And here for carrying out sequence analysis, sequence alignment, we have tools like BLAST, FASTA, dot matcher. These are the tools which are used for alignment of a pair of sequences, and we have tools like Clustal W, Clustal Omega, right, which are mainly meant for multiple sequence alignment. And we do have tools like Mega, Philip, and then POP, which are used for constructing the phylogenetic trees or phylogram. So phylogenetic tree, it is defined as pictorial or diagrammatic representation which deals with the relationship between the ancestors and the descendants. Ancestors and the descendants. So these are the four important modules in this so-called Biosuite. The first one is sequence and genome analysis. Second one is structural analysis. Third is simulation, and fourth module is the drug design. So all these four modules together, they constitute the Biosuite. And next, Biosuite, which is a comprehensive package of software tools, which has four important modules, which include sequence analysis, next building structure, carrying out simulations, and the fourth module is drug design. So here, Biosuite is nothing but a comprehensive package covering much of the activities of bioinformatics spectrum, starting from genome sequence to individual and multiple protein sequences, different levels of structure prediction. We have four levels of protein structures, that is primary structure, secondary structure, tertiary structure, and then quaternary structure. So we do have tools in the field of bioinformatics which are mainly meant for predicting the primary structure, secondary structure, tertiary structure, and then the quaternary structure, and later followed by the analysis of structures, molecular mechanics calculations, molecular dynamics, and then cheminformatics, and finally integration with the application of sequence and structural analysis in rational drug design through the algorithms for QSAR, which stands for quantitative structure activity relationship, identification of pharmacophore, and docking process for facilitating rational drug design. So Biosuite is nothing but a comprehensive package which covers much of the activities which are being carried out in the field of bioinformatics, starting from genome sequences. So we have databases in the field of bioinformatics which are mainly meant for maintaining this genome sequence of various organisms. To mention, we have a database for human genome, right? Similarly, we do have databases for maintaining the genomes of bacteria, viruses, plant and other organisms, and followed by genome sequences to individual and multiple protein sequences, at various levels. So protein exists in four conformations: primary, secondary, tertiary, and then quaternary, and followed by analysis of structures. So generally here protein structure is being elucidated through two important techniques like two-dimensional X-ray crystallography, and second is NMR, which stands for nuclear magnetic resonance spectroscopy. So these are the two powerful techniques which are used for elucidating the structure of the protein, which is basically a biological macromolecule. And we do have tools in the field of bioinformatics which are used for building the structure of the protein whose sequence is known but structure is unknown. So we call that molecular modeling. And coming to the tools which are used for performing this molecular modeling are Swiss Modeler, EasyPred 3D, and then iTaser, right? Later molecular mechanics and calculations. So especially we do have tools where we can perform this molecular mechanics and calculations and followed by molecular dynamics. And cheminformatics is a field, is a branch of science which deals with this pharmacogenomics. And in this cheminformatics, we do find the structures for the compounds which can be used as the lead or drug molecules. And finally, integration with application of sequence and structural analysis, especially in the drug design, through the algorithms of QSAR studies, quantitative structure activity relationship, and then identification of pharmacophore, and finally docking. So docking is defined as the interaction between the receptor protein and then the ligand molecule, right? So these are all the aspects which are covered. These are all the aspects which are covered in this so-called package Biosuite. And coming to the how Biosuite is organized. So it is very clearly organized into 4 important modules. The first one is sequence and genome analysis. The second is structural analysis, and third is simulations, and fourth is drug design. So Biosuite is organized into 4 modules. First module is sequence and genome analysis. Second module is structural analysis. Third module is simulations, and fourth module is drug design. So coming to this first module, that is sequence and genome analysis, right? So here coming to the sequence, here we have 2 types of sequences, that is nucleic acids and then amino acids. So genome sequence assembly and EST. So EST stands for expressed sequence tags, and ePCR, which stands for polymerase chain reaction, and then ORF prediction, open reading frames, and then intron exon boundary. So when you take the genome of either a prokaryote or eukaryote, it basically consists of, so genome basically consists of 2 components. First one is intron and second one is exon. So introns and exons, where intron is non-coding part and exonic part is the coding part. So coming to the prokaryotes, where genome size is small, we do have only exonic part. And coming to this eukaryotes, where the genome size is big, so we have both intronic part and then exonic part, where intronic part doesn't code for anything and exonic part codes for all the things. And next is database search. So once database is created, then we do have tools in the field of bioinformatics where we can retrieve the data from the databases, and later we can perform sequence alignment. So here in this sequence alignments, we have pairwise alignment, multiple sequence alignment, and then whole genome alignments. So here we need to discuss about the concept, that is dynamic programming. So dynamic programming, it is defined as alignment of a pair of sequences either globally or locally using Needleman-Wunsch algorithm, either globally or locally using Needleman-Wunsch algorithm or Smith-Waterman algorithm is called as dynamic programming. So the definition of dynamic programming reveals us there are 2 types of alignments, that is global alignment and the local alignments. Global alignment is nothing but alignment of a pair of sequences along the entire length, and the algorithm used for global alignment is Needleman-Wunsch algorithm. And the second alignment, that is local alignment, alignment of a pair of sequences along the conserved regions, and the algorithm uses Smith-Waterman algorithm. Here, when the number of sequences are 2, when n is equivalent to 2, we call it as pairwise alignment. When n is greater than 2, we call it as multiple sequence alignment, right? And we have tools for carrying out pairwise alignment. The best example is BLAST, which stands for Basic Local Alignment Search Tool. And for carrying out this multiple sequence alignment, so we have tools like Clustal W, Clustal Omega, which are mainly meant for carrying out this multiple sequence alignment. And coming to these motifs and patterns. So these motifs are nothing but conserved regions. These are nothing but conserved regions. And next is RNA and protein structure and transmembrane prediction. So here protein exists in 4 different conformations: primary, secondary, tertiary and then quaternary. So we do have tools in the field of bioinformatics, especially for finding out the primary structure, secondary structure, tertiary structure and then quaternary structure. So coming to this primary structure, so primary structure of protein is nothing but linear arrangement of amino acids one after the another through the peptide bond. And coming to the secondary structure of a protein, which basically consists of helices, sheets, turns, loops and then coils. Coming to the helices, it is again of 2 types: left-handed alpha helix and then right-handed alpha helix. Coming to the beta pleated sheets, they may be parallel beta pleated sheets or anti-parallel beta pleated sheets. And phylogenetic analysis is performed by using tools like MEGA, which stands for Molecular Evolutionary Genetic Analysis Tool, or we can also use the tool PhyLip or POP. So what is this phylogenetic tree? So phylogenetic tree is nothing but a tree-like or graphical representation which depicts the relationship between the ancestors and the descendants. So from the phylogenetic tree, we can just derive the ancestor and then the descendants. This is all about the first module, that is sequence and genome analysis. So coming to the second module of Biosuite, that is structural analysis. So here the basic biological macromolecules which are nothing but the nucleic acids. So if you just see the structure of this DNA, which is a double helical model, and coming to this protein which exists in various conformations, and these can be found out by using this module, that is structural analysis. And we can also perform this homology modeling and then ready. So homology modeling, which is also called as comparative modeling or molecular modeling. Here in this comparative modeling or homology modeling or molecular modeling, it consists of 6 important steps, right? First step is template selection, second step is target template alignment, and third step is building model framework, fourth step is adding loops and side chains, fifth step is model optimization, and the sixth step is model evaluation. So coming to this homology modeling, it consists of 6 important steps. First step is template selection, second step is target template alignment. Once template is selected, next thing is you need to find out the suitable target. Third step is building model framework. Fourth step is adding loops and side chains, and fifth step is model optimization, and the last step is model evaluation. So these are the 6 important steps in this homology modeling. And coming to this third module, which is nothing but simulation. So here, whenever we try to build a molecule using this molecular modeling or homology modeling or comparative modeling, so the model which is built, it should have minimum energy. So as we all know that molecules with high energy, they are always unstable and they try to get stability by losing energy. And we do have energy minimization tools in the field of bioinformatics, which mainly includes AMBER, CHARM and GROMACS. So these are the bioinformatics tools or computational tools which are used for energy minimization, which are used for carrying out the energy minimization. And we do have energy minimization algorithms like steepest descent algorithm, conjugate gradient algorithm, and we have this force field, right? Here force field, which is nothing but represented in the form of mathematical equation, that is total energy U, which is nothing but energy because of the bonded interactions as well as the energy because of non-bonded interactions. So coming to this term, energy because of bonded interactions, which mainly includes energy because of bond length, energy because of bond angle, and energy because of torsion. And coming to this energy because of non-bonded interactions, which mainly includes energy because of van der Waals forces, energy because of Coulombic interactions, and energy because of Lennard-Jones potential. So, if you represent this in the form of a mathematical equation, that is nothing but the force field. That is nothing but the concept of force field. And we do come across this RMSD values, root, mean square, deviation, and these all things. And we are also going to plot this R plot. It's also called as Ramachandran plot, and which is mainly used for predicting the secondary structure of the protein. So, all these things we do carry out in this third module of Biosuite, that is simulation. And coming to the fourth module of Biosuite, that is drug design. And here we have drug design approaches, that is structure based drug design and then ligand based drug design. Structure based drug design and then ligand based drug design. And conformation search, steric and electrostatic ligand alignment, and QSAR studies, quantitative structure activity relationship studies, and then pharmacophore identification and then pharmacophore based search. So, these are the four important modules of the so called Biosuite. So, coming to the first module of Biosuite, that is sequence and genome analysis, it is the first module of the Biosuite. So, this module, it mainly deals with the applications relating to the analysis of nucleic acid and protein sequences. So, as mentioned earlier, there are two types of sequences. First one is nucleic acid sequence and then the protein sequence. Not only for individual molecules, but also of complete genome and proteome sequences. So, complete genome means all genes, proteome means all proteins, and this module, it will enable researchers to annotate genomes of organisms, predict protein secondary structures. We have tools like Ramachandran plot, which is used for predicting the secondary structure of protein, and compare two genomes for similarities at the gene or protein level. So, we have this genomics. Genomics is divided into three components: structural genomics, functional genomics, and then comparative genomics. So, in the field of comparative genomics, we are trying to compare the genomes of organisms, different organisms, so that we will see how much amount of the genome is conserved between the two organisms. And this module is further divided into four sub-modules: sequence analysis, of course, genome analysis, comparative genomics, and then utilities. So, this first module, that is sequence and genome analysis, it is subdivided into four sub-modules. First one is performing sequence analysis, second is genome analysis, third one is comparative genomics, and fourth sub-module is the utilities. So, sequence analysis of individual molecules is enabled through sequence analysis modules, while the programs in the genome analysis, we can carry out the genome analysis through this first module, that is, and the sub-module enables comparison and analysis of full genomes and full proteomes of different organisms. And here database searching tools, BLAST, which stands for Basic Local Alignment Search Tool, which is mainly meant for alignment of a pair of sequences, and the type of alignment is local alignment. And another one is SciBLAST, and these are interfaced within the suite, that is Biosuite. And these tools, they will enable searching database to identify a given sequence or find conserved domains, or it even find distant related homologs from some other species. And the second thing is alignment of sequences, that is either global alignment or local alignment, which is a very important task in sequence analysis, is provided through two well established algorithms, that is global, and the algorithm is Needleman-Wunsch algorithm, and local alignment, that is Smith-Waterman algorithm. So, we can perform two alignments, that is global alignment by using Needleman-Wunsch algorithm, and we can perform local alignment using the Smith-Waterman algorithm. And coming to this sequence and genome analysis, here the algorithms, they are also there for secondary structure prediction of protein and transmembrane region detection, RNA structure prediction, and all these are part of this module. So, for predicting the secondary structure of protein, we have tools like SOPMA, GAR, NN Predict. There are many tools in the field of bioinformatics which are mainly meant for predicting the secondary structure of the protein. And this module, especially this first module, it has programs for studying molecular evolution. That is nothing but we can do this by using tools like Mega and then Phylum. Right. So, and this module, it also has algorithms for finding of genes, that is ORFs, open reading frames, gene assemblies, and then we can also design primer, and then EST, expressed sequence tags analysis. So, these are all the things which we can do in this Biosuite first module. And here we can clearly see the tool which, especially this sequence and then genome analysis, which is being carried out by using the tools which are available in the first module of Biosuite 1, first module of Biosuite. Right. So, here you can see how a phylogram is constructed here. It is nothing but phylogenetic tree. You can clearly see how phylogram is constructed, and we can use tool like Mega, molecular evolutionary genetic analysis tool, which is used for constructing the phylogram. So, phylogram helps us in revealing the evolutionary relationship between the ancestors and the descendants, So, this is a rooted tree where the ancestor is known, and we have many nodes. Node is nothing but point of branching or bifurcation, and we have many operational taxonomic units, right. So, here the first output is nothing but genome comparison, mapping protein from Bacillus halodurians to cluster orthologous groups, and coming to this second, is nothing but a tool which is meant for predicting the secondary structural components of a protein, right. As mentioned earlier, protein exists in different conformations. So, protein exists in 4 conformations: primary structure, secondary structure, tertiary structure, and then the quaternary structure. So, here we can use tools like SOPMA, GAR, NN predict, right. So, these are the tools which are used for predicting the secondary structural components of a protein. So, coming to the secondary structure of protein, it mainly consists of helices, sheets, turns, loops, and then coils. These are the components of secondary structure of a protein. Helices, again there are of 2 types, right, alpha helix, right handed alpha helix, and then beta helix. And coming to this beta pleated sheets, they may be parallel or anti-parallel, Coming to the second module of this Biosuite, which is used for modeling, 3-dimensional modeling, and then followed by analysis. 3-dimensional. So, the 3D modeling and analysis module, it has capability to build the structure for a protein whose sequence is known, but structure is unknown. So, here we are trying to build a structure, we are able to predict 3-dimensional structure for a macromolecule, that is protein, through this molecular modeling. So, this module, the second module, it is again further subdivided into sub-modules like homology modeling. So, in this homology modeling, we are trying to build a model for a protein whose sequence is known, but structure is unknown. So, here in this process, we are trying to compare protein with both structure and sequence known to our protein whose sequence alone is known, but structure is unknown. So, we are trying to build a structure for our protein basing on the similarity of our protein to the protein in the database. And the threading is also used for building the structures and building proteins, building nucleic acids like DNA and RNA, building carbohydrates, maybe monosaccharides, disaccharides or polysaccharides, may pentoses or exoses, generation of symmetry related molecules, structural superposition, surface and volumes, and we can also predict the binding sites. And there is one tool by name CASP-P, which is mainly meant for finding out this active sites, and nucleic acid analysis, and we can also see the interaction between the atoms in a molecule, and we can also do this quality check and fold detection. So, building the models of protein molecules by predicting their three-dimensional structure by comparative modeling technique and enabled through the first two submodules. So, here the first two submodules are nothing but homology, modeling and threading. So, these are the two submodules which are mainly meant for building the structure of a protein through the technique comparative modeling, homology modeling or molecular modeling. So, building nucleic acids, carbohydrates using geometric information, it is enabled through the building modules. Here we are able to build nucleic acids, structures of nucleic acid, and at the same time structures of carbohydrates, maybe pentose sugar, hexose sugar, and maybe monosaccharide, disaccharide or polysaccharide we are using through building modules. And there are several programs that can address visualization and analysis of crystallographically derived structures, which are also included in this module. Here RASMOL is one tool which is mainly meant for visualizing the three-dimensional structures of biological macromolecule like protein. And we do have algorithms for classifying protein structures as primary, secondary, tertiary and quaternary in relation to the other protein structure which are known from the literature database. And we do have a database for protein structure classification, that is CAT, which stands for class, architecture, topology and then homology. These are the four levels of protein structure classification, and we have a database by name SCOP, structural classification of proteins. And next, structure validation tools, especially for proteins and nucleic acids, they are which together constitute the biological macromolecules. They are enabled through quality check programs, and extensive analysis is possible through the analysis and interaction functions which can be used for analyzing integral features of protein structure as four conformations: primary, secondary, tertiary and then quaternary, and then protein-protein interactions as well as protein-ligand interactions which we come across in docking. So, docking is a process where we do find interaction between the receptor protein and then the ligand molecule. So, here we can see how the interaction happens between the receptor protein and then the ligand molecules. And on the right hand side we can see the Ramachandran plot. This is nothing but the Ramachandran plot which consists of four quadrants, and it is plotted between the psi and phi angles which range from -180 to +180, and it is mainly meant for predicting the secondary structure, protein secondary structure, which consists of helices, sheets, turns, loops and then coils. So, on the third important module of this comprehensive bioinformatics software tool, that is Biosuite, where we do carry out the simulations, and it is possible through this module, that is third module of the Biosuite, where we come across the concept of force field, which is represented in the form of a mathematical equation. And then we do have energy minimization tools like GROMAX and then AMBER and CHARM, and we can also perform this Monte Carlo simulations and then electrostatics. So, here when you come into the force field, which can be represented in the form of a mathematical equation, U total is equivalent to U because of bonded interactions and U because of non-bonded interactions. So, coming to these bonded interactions may be due to bond length or bond angle and then torsion, and the non-bonded which is basically because of this van der Waals interactions, Lennard-Jones potential and then Coulombic interactions. And here we do find several algorithms for first order unconstrained energy minimization. As mentioned earlier, we have energy minimization algorithms, and then we have energy minimization methods, and we have energy minimization tools, especially GROMAX and then AMBER and then CHARM. So, these are the tools which are mainly used for carrying out this energy minimization. So, why do we need to do this energy minimization? So, whenever we try to build a structure for a protein using the concept of molecular modeling. So once the structure is built, so we need to see whether the structure which is built, which is of having high energy or low energy. Suppose if the model, suppose if the structure is built with high energy, so it is always unstable, right? So we need to perform this energy minimization so that the built structure will get the stability. So that is done through this energy minimization. Here we can clearly see how energy minimization is being done. Energy which is represented in the form of kilojoule per mole or kilocalorie per mole, and then versus the different temperatures, and then time. You can see how energy minimization is being done. So molecules with high energy, they are always unstable and they try to get stability. So in this process, they do lose some energy, right? Losing of energy, which is termed as energy minimization. Energy minimization, right? Coming to the last module of this Biosuite, that is drug design. So nothing but it can be done through the concept of docking. So docking is simply defined as the interaction between the receptor protein and then the ligand molecule. Here, receptor protein and ligand, they try to interact with each other in different conformations, and for every conformation there will be some energy, right? So in whichever conformation both this receptor protein and ligand molecule orient themselves, right, in whichever conformation it has least energy, that is going to be the most stable form. And here this module, it provides the following functionalities, that is prediction of biological activities of unknown chemical entities using QSAR studies, quantitative structure activity relationship, and then identification of pharmacologically biological active molecules, and then superimposition of set of molecules in three-dimensional space by alignment, and then identification of ligand poses in 3D space when it binds to the target using docking. And docking is performed by using tools like Autodock, Vena, and then Gold. Right. And here lead optimization, it can be achieved iteratively if the target structure is known, then the lead optimization can be done using the structure based drug design such as by docking. So here the process of alignment of a set of molecules in three-dimensional space to find the superimposable regions of a group of molecules or to estimate molecular similarity, it can be performed by using the concept of field fitting. So, and for deriving and matching 3D pharmacophores, the following features are extracted or used. The first one is hydrogen bond donor, second one is hydrogen bond acceptor, third is aliphatic hydrophobic group, fourth one is aromatic ring, and fifth one is negatively charged groups, which is nothing but anions, and then positively charged group, that is nothing but the cations. And finally, creating and refining a training set required for quantitative structure activity relationship predictions. They are aided by means of k-means clustering, k nearest neighbors, and then UPGMA hierarchical clustering algorithms, where UPGMA stands for unweighted pair group method using arithmetic. And here we can clearly see the interaction between the receptor protein and then ligand, and finally this is the best fit where the energy is minimum. And coming to the summary of this topic, that is Biosuite. So Biosuite is nothing but comprehensive bioinformatics software package which was developed between a unique industry academia interaction, that is taken up by CSIR and then TCS. And this so-called Biosuite, it has 4 major modules as discussed earlier. The first one is genome and proteome sequence analysis. Second module is 3D modeling and structural analysis. Third module is molecular dynamics and simulation. And the last module, that is drug design. So the possible questions in this topic. So here the student is expected to know about student is expected to know about this concept of Biosuite in detail, and at the same time the student is expected to know about the 4 important major modules and again sub modules. So each major module is again subdivided into sub modules. So, these are all about the possible questions in this topic, and these are the references, standard books for this molecular modeling by R. Leach. These are the standard books. Thank you.